So I know I've given you lots of genetics problems and we worked through lots of examples, but let's just go through a little bit of your genetics experiment just to make sure everybody's on the right page here. So first of all, we need to recognize that capital Y, which means it's dominant, is the gene for the yellow or the allele for the yellow phenotype. Little y is the allele for the blue phenotype. So it's asking us to do a Punnett square for two different examples. Okay, so we will do just that. Create a little Punnett square here. So let's do this one first. Okay, so we'll create a Punnett square here. One parent, right? So this portion up here represents the gametes that are possible for that parent. That parent only has one allele, right, that can be a possible gamete. Okay, the other parent actually has two different alleles for each of their gametes. So then we just fill in, carry this across, carry these down. Okay, and this would be what's represented inside the Punnett square. First of all, it tells us what are the possible genotypes for the offspring, and then it also tells us what the probability is. That doesn't mean, right, that you will get exactly that, because the probability that I flip a coin and get a heads is 50%, but I might flip heads twice in a row, right? It's just telling me the probability. However, I know that if I flip a coin, I'm going to get heads or tails. I'm not going to get something else. And so the genotypes inside these squares tell you what are possible. And then we can determine the phenotype from those genotypes. So as we see, we have a dominant allele, which is capital Y, and we have a recessive allele, which is lower y. So if one capital Y allele is present, then we know that it's going to express that phenotype. So we see that 100% of these offspring are going to be yellow based on the outcome of our Punnett square. Are there any blue kernels? No, right? Because every single possibility is going to have at least one of the dominant alleles. Okay, how can we tell whether or not there are blue kernels? Well, we know that Y is completely dominant to little y, therefore, in this case, there will be no blue kernels. Now let's do the other example. It's important too to remember the difference between phenotype and genotype. So genotype means the gene sequence. We represent that by these letters, capital Y, little y. Phenotype is the physical appearance, like yellow kernels or blue kernels. So now we're going to do this example, okay, for our Punnett square. So one parent only has the dominant form. So this is a homozygous dominant parent. This is a homozygous recessive parent. So we see that with this cross, 100% of their children will be heterozygous, is that will be their genotype, big Y, little y, and 100% will have yellow kernels. That's their phenotype. Okay, so the next question says they want us to set up a Punnett square and cross 2 of the F1 from cross 1b. So this is cross 1b that we just performed here, right? This is P and this is P, so these are the parent generation. F1 means the first filial generation, so this is what we have just figured out is what F1 is. So they want us to cross 2 from the F1 generation, so that would look like this. Big Y, little y crossed with big Y, little y. Okay, and they want us to see what the resulting Punnett square looks like from that cross. So this would be what we would call a mono-hybrid cross. Mono meaning one trait. Hybrid meaning both of the parents, okay, are heterozygous or hybrid for that trait. So each parent can form a gamete of either the homozygous or the heterozygous, the homozygous, the dominant or the recessive allele, And we'll fill in our Punnett square. And we see that the genotypic ratio is 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Our phenotypic ratio would be 3 yellow kerneled, right? They're phenotypically there, you wouldn't see the difference between these, and 1 blue kerneled offspring, or 75%, 25%. Now, if we look at number 5, this is an example of a genotype where there are actually 2 traits, not just 1. And so it's a little more involved to figure out all possible gametes when you're talking about 2 traits. Okay? And so you can use a method called foiling if you want to, or you could just figure out all possible combinations, assuming that these are not linked genes. So in this first example, okay, we can have a combination of big Y with big S. Okay? We can have a combination with big Y with little s. And then big Y, big S again, right? This would be a repeat. And the same thing here. Big Y with little s. Those are all the possible gametes for that particular organism. Okay? Now I'm going to go ahead and do it's asking me to make a Punnett square using these gametes as P. Okay? So in other words, we're going to use this cross. Okay? When you have 2 traits, your Punnett square gets quite a bit bigger. Okay, so we would have 4 possible gametes. Okay, and with 2 traits, remember the gametes, like a sperm and egg, are going to have half the genetic material. So since we had 4 letters or 2 genes, then we're going to have 2 letters, 1 for each gene in our gamete. So we have a possibility of big Y, big S, big Y, little s, big Y, big S, big Y, little s. And we have the same thing here. Now, you can fill in the entire 16 boxes for this Punnett square, or you can recognize that these 2, right, are repeats of these, and so we could just ignore that portion, right, because these are not any different than these 2. And the same thing here. We could ignore this because these 2 are repeats of these 2. So really the only possible gametes are just either big Y, big S, big Y, little s. That's it. So we can fill those in. And see our genotypic and phenotypic ratios. So we have 1 that's homozygous dominant, right, or 25%. We have 2 that are homozygous dominant for the color of the kernel, and heterozygous for wrinkled or smooth. And then we have 1, or 25%, that's homozygous for the color, homozygous dominant for the color, and homozygous recessive for the wrinkled or smooth. Now in the next part of your experiment, you are basically randomly choosing what your parents would look like based on putting colored beads into a beaker and pulling out combinations of those colored beads so that you could set up different parental crosses. Okay, so for example, if you put 50 blue beads in and 50 yellow beads, then when you pull them out, your first combination, might have been that you got the yellow, and the next one you might have gotten a blue bead, and then you could do that cross. Okay, so oops, sorry. It just depends on the way that you draw out your sequences. And the next time you draw the beads out, you might get 1 of each. Okay, you're just setting up all different possible crosses, and then you can actually set up a Punnett square for those crosses to determine what their offspring would look like. In the next part of the experiment, you're simulating a dihybrid cross, and you're going to need two different beakers and two sets of colors. So you could use blue and yellow beads for one gene to represent the two different allele types, and then maybe red and green beads for the next type of cross. And that completes your lab activity for genetics.